So I'll go through my experience making E. coli lysate systems and just sort of point out the main things that I think you should take into account when you're also trying to make these systems. So I'll just reiterate again the benefits of using cell-free systems. So if you're working in vivo, it's nice because you automatically have the transcription translation machinery readily available. However, you don't know if there might be some interactions between the sort of synthetic gene circuit you've introduced and the cellular environment. You also have to deal with the fact that the cell is growing and dividing as well. So that might interfere with your synthetic gene circuits. And then the most critical disadvantage, I think, is that you have to spend a lot of time cloning all the different gene constructs you have into the cells. So you can spend a lot of time doing that. But instead, you could use cell-free systems. So you basically take out all of the protein components from the cell. So you have this transcription translation machinery. You combine it with an energy solution, and then you can produce the proteins in vitro. And it's a nice open and controllable system, and you can use linear DNA templates, so you don't need to clone so many things. So I'll try and give you guys an overview of everything that is included in these E. coli lysate extract systems. So it's composed of the lysate, which is the protein components from the cell. And then you have your energy solution with the NTP, salts, cofactors, amino acids, and some form of ATP regeneration. And then you also have the DNA templates. And I would encourage that you use linear, but as it's iGEM, you also get your plates with different plasmids. So if you have something that you want to test, you can also use plasmids. It works also well. And yeah, the resources I have on this page are just the publications that I used to create my protocol, and they're very helpful also, I think, for everyone starting to work with E. coli lysate systems. So this is sort of my map of making lysate. So I'll just go through the general steps. The first day, you start with your mini culture. So it's just a 5 ml culture with a certain bacteria strain. In my case, I was using E. coli. And then you take some part of this culture after growing it overnight and you inoculate larger culture volumes. So I was typically doing 4 times 200 ml, but you can always scale this depending on what you need and what you want. And then after you grow these to a certain amount, you then harvest them, spin the cells down, and then you wash them a few times. And then once you've washed to remove all of the media, you can then freeze dry your cell pellet. And then technically you can continue on to the 3rd day without like freezing in -80. But I think actually the freeze drying of your pellet helps it to then lyse easier. So normally I break it into 3 days. And I think maybe if you're starting out new, it's also easy to separate because the first time you do all the washing, it's kind of tedious, so you might be tired. And then on the 3rd day, you resuspend your cells in a smaller volume. Normally it's a 1-to-1 ratio of the grams to milliliters of your wash buffer solution. You then lyse the cells. I was using mostly sonication, and there's a nice protocol by Quan and Scientific Reports that highlights exactly the parameters for sonication. So if you have access to that, I think it's a very nice way to do it. You then centrifuge your lysate, and then you remove the supernatant. you can do a runoff reaction at 37 degrees Celsius in order to eat up all of the genomic DNA present in your lysate. Then you centrifuge again, and then if you need to, you can do dialysis or buffer exchange. So there's a few things that I think are maybe important to keep in mind. And the first day is mostly what cell strain do you want to pick and what type of medium do you want to use. So the cell strain, depending on what you want to do, maybe the easiest is using BL21 strain, because there's a lot of examples of this in literature, so you have a lot of protocols to sort of troubleshoot with. And also if you use BL21DE3, then you also have the possibility to induce the expression of T7 RNA polymerase, so that's nice if you want to work with that RNA polymerase rather than the E. coli holoenzyme. And then depending what types of proteins you want to express, maybe you need a different strain that doesn't have a certain protein because you want to express it. And yeah, the medium is important because if you want to make a simplified energy solution, which I'll talk about a bit more later, then you have to use a medium with phosphates inside so that you can use a more simple energy solution. So just to go in more detail about this energy solution. So the main protocol, which a lot of people are using, is the one from Zach Sun in Jove in 2013. And he uses a more intensive energy solution, which costs more money and has more components, but it works very fast and also very well. Otherwise, I refer you to this paper by Kai et al. In 2015, who made a more simplified energy solution with 12 components, and it costs a lot less money. The main difference is that in the Sun paper, they use NTPs and a 3-PGA as the energy source, so the kinetics are different, and you can see that with this first energy solution, the reaction happens much quicker. And if you use the simplified one, then you basically start from NMPs, and then based on how you grow the cells, you have the machinery that produces the NTPs for you. So it takes a bit longer because you're producing at first the NTPs, and then the reaction starts going afterwards. But the main thing to see is that if you do LB and you try to use the simplified energy solution, it definitely doesn't work. So you have to take that into account if you want to use that energy solution. On the second day, there's maybe a couple of things to consider, mostly what sort of optical density do you want to grow the cells to before you harvest them, and which wash buffer should you use. And in the case of optical density, I've seen a lot of different things that are in the literature. When I first started making it, I thought it was really important to do it to harvest the cells in the exponential growth phase. However, then I started realizing that people do all sorts of stuff, and in my experience, it hasn't made too much of a difference, like in terms of what I was doing, but perhaps you should just take note of it because it could affect your experiments depending what you're doing. And then depending on if you do dialysis or buffer exchange, you maybe need to use a different wash buffer. And then on the third day, there's a number of things that are nice to keep in mind. So maybe when you first start with the lysis, you maybe need to know what method you will use. As I said before, the sonication is nice because Quan described very well the parameters to use for sonication. but I know that you can also use French press. I personally had some problems with bead beating, but depending if you have like a bead beating machine that has different settings, I think it's okay. Or you can do chemical methods, or there's a lot of opportunity depending what you have available to you. And the thing that I always like to see is that when you, like before you lyse the cells, it will be like very opaque, thick solution, and then afterwards it should be more transparent. And then when you centrifuge this cell lysate solution down, you should get 3 clear layers, 2 layers in the pellet, and then clear transparent supernatant above, which is what you take to do the runoff reaction. And I think some people say this is optional as well, the runoff, but I always do it and it seems to be useful. And then you centrifuge again, and then depending if you need to or not, you can do dialysis or buffer exchange, which can be important depending on the cell strain that you have. And then, yeah, you can use just the standard dialysis tubing, but sometimes sometimes it's better if you have larger volumes for that, depending on the size of the tubing. So I also tried these Slide-Alyzer mini dialysis units, which are very good for small volumes if you're trying different parameters out. And then you could also, instead of doing dialysis, do just a buffer exchange with the centrifuge filters, which also works. So just as an example for this, because originally when I did, when I was making lysate, I just was doing BL21 strain. I was making from BL21 strains, and without doing dialysis or buffer exchange, it was absolutely fine. But then for my experiments, I wanted a lysate without a LAC inhibitor present. So I tried to make lysate from 2 different strains. And I found in this case, for one of the cases, it was very important to do the dialysis actually, because yeah, as you see, with just the runoff reaction or without the runoff reaction and no dialysis, the expression is either very strange or not at all. Only when I do sort of the real dialysis even is better than also just the buffer exchange. But then if you look at one of the strains, the MC4100, it's completely different. So only without the runoff, it doesn't work. So in this case, I find the runoff to be also useful. And then when you go on to test your lysate, you can do it with either plasmid or linear DNA. With plasmid, since it's circular, it's already protected naturally against the nucleases that will be in your lysate. But if you want to use linear templates, then you do also need to add something extra to protect it from the nucleases. So you can either add this GAM-S protein, which protects the linear DNA templates, or you can add a DNA which contains a what's called a chi protection sequence, and that also protects your linear DNA template. So the chi sequence is easy because you just anneal 2 DNA oligos together, and it's very fast to do. However, if you want to do the GAM-S way, it's also not that difficult. You can either add the purified GAM-S protein. Otherwise, you can also mix lysates together. So in this case, we mixed a lysate we made from top 10 cells. And in these top 10 cells, we induced the expression of GAM-S. So we could mix the lysate that has GAM-S in it with a BL21 lysate, and actually it works quite well, sort of regardless of the ratio. It works well. And yes, so in conclusion, I hope that you maybe are convinced that cell-free will be useful to you, and you can save a lot of time and avoid cloning to test linear DNA constructs. And I think since everyone in iGEM is working in teams, it's actually not very difficult to make the lysate and work together. And there's a lot of resources. So I think it's maybe annoying the first time, but I think together with a team, it's not that difficult. And also another thing to consider is if you really do want, for some reason, things to work in cells in the end, it's actually okay, because you can also just use this as a prototyping system, and then it should translate as well to working in vivo afterwards. Okay. Thank you, and good luck on your project.
Zoe, do you want to answer some of the questions before you move?
Oh yeah, I don't know.
So there was one question, I will start with the later one. Which strains are common for such a process? So you talked about it, but maybe you can summarize just quickly.
Yeah, I think the most common is these BL21 strains. I don't know if I've seen other ones, but I think
often was also A19 use, but I think it moved a lot more to the BL21.
Okay, yeah, because I guess like both of the protocols that I'm citing, they are using BL21. So, but yeah, depending what you want to do, like sometimes if you're going from a certain paper and you want to try and replicate what they did, then it's also important to take into account the strain they used, I guess.
Yeah, and then there was a question about what is more efficient if cell-free or in vivo protein synthesis. For example, in protein yield, regarding protein yield.
I guess the lysate is still, I mean, I'm not exactly 100% sure how much better pure it's gotten now, but I guess it's still more efficient, the lysate, no?
Yeah, so yeah, but I think it was in vivo comparison with like a cell expression. So in that case, I would say it's more about if you are doing something, there are like different advantages to using cell-free than protein yield, but otherwise protein yield is much better in cells. So if you are just trying to purify protein or something that you can easily do in E. coli, then it's better. Do you agree?
Yeah, I agree. I think it's only useful if you're doing something that is like toxic to the cells, or or you have to like really control the system more or something.
It also depends on the size of the batch where you're running your reaction, and you can produce much more protein at a larger scale in bigger batches, and they do that industrially. But I think it's usually for more technical constraints that you would decide to pick cell-free, and especially if the protein is from the host or from the organism or not, you'd always get a better yield in vivo, I guess. But it's more, I don't think you should pick one or the other because you want to get a higher yield. I think it's more about the constraints that you have in your system. And for example, you can have different constraints. You can have biochemical constraints, like we were talking about toxicity, but you could also have time constraints. And once you've spent many hours preparing your lysate, you can keep it, you can freeze it, and then you can reuse it, and then you can work really fast. Like you have your little aliquots, and you can just reuse them over and over again to just prototype your work. So I think it's got some advantage compared to vivo.
Okay, there was additional questions for this. So for cost effectiveness, I should choose in vivo. So I guess this is what we were talking. It's very complex, not so straightforward answer. So it really depends what you are doing.
Yeah. Also, I guess like for iGEM, I don't see a huge need to like really scale up reactions to like a huge extent. So in the case of iGEM, I think it's very useful because I don't think anyone needs to make like grams of protein or something like this. So I think
yeah, exactly. For the competition, for example, if you manage to express the protein, I think, you know, the judges would be really happy to just see that. And if you can just prove that, you don't need to you know, you're not expected to produce something as some industry would do. Or,
yeah, I mean, it's good to think about, but in that sense, it's like, since it's only the beginning with iGEM, like it's only the beginning of a project. So it's very nice because you could potentially try much more things using cell-free than you could if you spent like all your time cloning, basically.
yeah.
Okay, so are we fine? Can we move on?
Yeah, let's move on to Barbara's talk.